Okay, let's get started. Everyone sufficiently caffeinated? It's good to be back in Milan. It's good to be back on the Human Cell Atlas meeting. It's by far my favorite meeting of the year. It's good to see so many friends, almost family and colleagues around, and many new members of the HCA community. Today, it is my honor to present a new program, the HCA Spatial program. So why a new program? Because we thought technology is ready now. It might not have been ready 2 years ago, but now it's really technology matured. We can be better in capturing genes in cells by imaging. We are better in capturing molecules in smaller units, like smaller spots or nanoballs. And now it's really the time to push Atlas efforts forward from dissociated single-cell level to spatial resolution. And that's obviously not a new idea, so it's not a new kid in the block, but it has been actually described in the very first white paper where spatial biology or the spatial technology was, besides single-cell RNA sequencing or single-cell omics, one of the pillars to generate atlases of the human system. However, so far we have been generating atlases mostly on single-cell level. That this has mainly been true or due to the fact that single-cell technologies had been a higher technology readiness level. So we have been able to generate large data sets cost efficiently, very robustly using single-cell technologies. But over the last, let's say, 2 years, we gained more experience and we have been much better generating also spatial atlases. At this point, more complementing what we've done on the single-cell side, kind of bringing what we learned from single-cell dissociated cells into spatial atlases. But now things changed quite a lot with technology for spatial omics profiling getting much better, much more precise, much more robust, cost efficient. We still have to see, but it's definitely in a place where we can use spatial omics to generate atlases, either in combination or using in combination with single-cell or just using spatial omics alone. And this brought us here. But as a program, obviously, we have a vision to push it forward and to move from the two-dimensional context, the way we are now profiling spatial biology, to a three-dimensional map of entire tissues and organs, but eventually bring it really to a four-dimensional atlases, integrating three-dimensional organ maps and mapping that over time, for example, during human development, but also during lifespan while we are aging. And this is really where we are today. So we have been generating a lot of data. We're standing on the shoulder of giant single-cell data sets. And these are then used now to complement what we have been doing on the spatial side. But the future really is using spatial biology and spatial technology alone to build atlases moving forward in two dimension, 3 dimension, and including temporal components moving forward. So what we don't want to do moving forward is to dissociate cells, to take cells out of their original context. And ideally, we don't want to slice cells. So spatial technology today is really based on the fact that tissue organs are sectioned and molecules are profiled within those sections. But moving forward, we want to be better and really appreciating the full complexity of transcriptomes or molecules within cells. So profiling cells in 3 dimensions and then even profiling tissues and organs in 3 dimensions. So as a program, we kind of designed 3 different tracks, where the tracks are basically split in 3 components. One is the technology, then the data analysis, and eventually the building of atlases in health, but also disease. So we won't be alone. So this is built on efforts we have been doing over the last 8 years now in different working groups, mainly the Standards and Technology Working Group, where we defined kind of standards, guidelines for mainly single-cell resolved methods, but then also obviously the Analysis Working Group and eventually the bionetworks will have a very important role driving now spatial technology into the next phase and into atlas building efforts. So here, what is happening today is that we have opening talks. So the first hour has been used to introduce technology, to introduce analysis pipelines, but then also some examples how spatial biology is already used to build atlases in health and disease. And then after this hour, we're breaking into 3 different breakout sessions, again focusing on technology, analysis, and atlas buildings. So these are the framing talks. So I will start giving a bit of an overview of the landscape of technologies that are today available to build spatially resolved atlases. And then Ollie, Kevin, and Grace will follow up, introducing the computational challenges and challenges to build atlases in health and disease. And these will then convert into the breakout sessions. In the breakout session, as a program, we aim to define priorities. So questions, challenges we want to tackle first, but also to work already on minimum viable products that we can use to showcase the power of spatial technologies in building next generation atlases. So with this, I will kick it off and give you a bit of an overview, a bit of a landscape of technologies that are out there. It's not meant to be comprehensive, and also it's not showing today benchmarking efforts, because we have been benchmarking a lot on the single cell side, but this is much more difficult to translate to spatial technologies. So what I will kind of present is split between technologies based on imaging and based on sequencing. So those technologies that capture molecules followed by sequencing, and those that image molecules directly within the tissue. One resulting in single cell resolution, the moment cell boundaries are well defined through cell segmentation, and the other providing kind of a pixel-based resolution. Today, we mainly build atlases based on RNA sequencing, but there has been a lot of discussion today already around additional modalities, and also the spatial technologies moved in recent years from RNA alone to different modalities, even in combination RNA and different measures. So again, this is not a kind of a comprehensive overview, but really a high-level landscape of technologies that are out there and can be used today to build atlases. So in the Standards and Technology Working Group, we have been very busy initially to generate kind of guidelines and standards to build high-quality atlases. So this has been possible because as a reference material, we could rely on cryopreserved PMCs. So these are very easy to freeze, aliquot, ship, and then implement in a standardized process to test technologies. This is far more difficult on the spatial biology side. So technologies are very different, just considering sequencing and imaging-based methods, but also kind of within the 2 components, it's much harder to standardize, for example, availability or making availability of molecules by capture or for probe hybridization. So what we'll do today is basically to generate a catalog to allow you to make informed decisions, to pick technologies to build atlases, and then to have kind of to bring that discussion into the different bionetworks to kind of help them building the next generation of atlases. So in the first part, I will focus on sequencing-based spatial indexing technologies, which are broadly split in different capture units. So we are probably all familiar with the spot-based methods like Visium or the initial spatial transcriptomic methods. But then over the years, there was a lot of innovation, especially on the kind of capture chemistry and capture size side, really allowing us to capture initially around 100 micron spot size, but now really going down to a subcellular level. So if you read this as a kind of scale clock, where initially using spatial transcriptomics, so this is the original spatial capture methods developed by the Lundenberg lab, we could capture around 100 micron spot areas, and then over the years, really innovation brought us to subcellular level, which to date the highest capture efficiency with stereoseq capturing on nanoboards around 250 nanometers. So this really allowed us to go from an area profile to a subcellular profile. And this is a review published in 22, and you see already there's a lot of, it's a moving target, and there's a lot of innovation being done in this space. And two methods I would like to highlight, which I think today are the highest resolution open source methods with OpenST and NovaST, which both reuse or reutilize the NovaSeq flow cell to turn or convert the clusters within the flow cell into capture probes. So these are methods that really capture molecules at high resolution and allow to generate subcellular profiles today on RNA, but for sure there's a lot of innovation being done there. As a commercialized method with similar resolution, we have StereoSeq. Again, these are nanoball capture probes with around 250 nanometer spot size. And again, there's a kind of high resolution capture of molecules. In this case, even scalable to larger area sizes. So the MGI flow cells are of different size and have been used even to capture a full monkey brain. So there's different flexibility in capture area here. On the Visium side, so this is kind of a development following the initial spatial transcriptomic methods from 100 microns, then initially to 50 micron, and now 2 micron resolution that really allowed us to move from area to subcellular profiles. And I think a unique feature here is the use of FFP material that really allowed us to also kind of appreciate larger clinical biobanks in this context with high quality gene expression profiles at the end of this process. Another interesting innovation where spots are not spotted on a slide, but kind of capture probes are bound to beads to capture molecules has been initially described as a slide-seq and now slide-seq 2, and now it's commercialized by Curio as Curio-Seeker. That really allowed us to kind of be flexible in the design of the beads or kind of generation of the beads with today a resolution of a 2 micron bead resolution in this case. For me, one of the more innovative approaches here is a kind of evolution of this technology where molecules are not captured on the beads, so it's not a diffusion onto the surface of the array, but here with a photocleavable linker, the molecules are actually diffused into the tissue, and this allows the tagging of cells and nuclei, which can then be brought into standard single-cell workflows. In this case, we have a real single-cell resolution because there's no sectioning or damaging of cells being involved. And then following single-cell sequencing, we can use the indexes that we diffused into the tissue initially to bring cells back into the spatial context. On the imaging side, here showcased for the original MERFresh technology, most of the methods are based on the fact that probes are hybridized to RNA molecules, and there are different forms of decoding where then these probes contain different indexes related to the gene of interest, and the complexity of these indexes is then used to scale the number of genes being profiled. And since 2016, we've seen a lot of innovation here, commercialized by, for example, these 3 companies. And here the difference really is the panel size, where now gene panels really grew from initially around 100 or 500 genes to up to full transcriptome level these days, really allowing, together with the cell segmentation achieved by imaging, to perform single-cell atlasing based on the fact that we profile molecules in segmented cells and generate almost unbiased transcriptome profiles per cell. On the multi-omic side, there are kind of endless opportunities these days. Some are easier to implement than others. Here just a few examples where RNA is combined with copy number variations. So in this case, the original Visium slide was used to capture molecules, but then through the mapping statistics across chromosomes, one could infer copy number variations, as has been previously done on single-cell level. So here they also used infer CNV to generate a for this cancer a kind of hierarchical clonal landscape, which then together with the spatial coordinates could be mapped to the tissue and, for example, ask questions about co-localization of immune cells with tumor cells and then the inhibitory activation effect of the tumor microenvironment within this tumor. On the other side, the sequencing library itself can be used or reutilized. So in this case, from the original SlideSeq library, the authors re-amplified TCR and BCR sequences that then, together with the spatial index, allowed the kind of mapping of clonotypes into the tissue and together with the gene expression information of the T cells and T cell phenotypes to draw further conclusions about the activity, for example, anti-tumor reactivity of those T cells based on the T cell receptor sequence and clonal expansion profile. So this is probably one of the most innovative and flexible systems where this comes out from Rong Fan's lab, a method called DBIT-seq, where molecules are flowed onto the tissue, so not captured on the array. And here, this flow can be kind of flexibly used to do RNA sequencing, but the authors really brought it to the extreme, combining methods like RNA together with ATAC-seq, RNA together with histone profiling. So this is by far what I've seen the most flexible methods. With, again, as a take-home message, there's no limitation anymore what multimodal features can be combined. It's just a matter of selecting well. And this is part of the spatial program and where we contribute really to look at the technology landscape, to compare, to combine, to provide guidelines for the biological networks, to find a technology that is fit for purpose, but then also to look at very specific questions and to look at this kind of catalog and to inform what is the best technology selection for a given project and for a given generation of tissue-specific atlases. And with this, I will stop the technology overview. I will hand over to Ollie, who will brief you about the analysis pipelines. After the session, again, we have breakout sessions starting, hopefully not that much delayed. And I will hand over to Ollie to take over from here. Thank you very much.
Not yet coming up. Great, excellent. So I'm really happy to follow on from Holger's presentation on the technology side and talking a little bit about analysis, how they are emerging already and where we stand as a community to do so. And I think what's perhaps different is that on the analytical side, there's a lot of flux in the movement. And so I really want to exemplify also where the new challenges, opportunities, which we'll discuss later on, where others and some bits we contributed to have made starts we should all committedly build on and extend further. So I think I just want to review here the basic workflow of analysis. I think much of that already been really exemplified by Holger's talk very well. From the imaging versus sequencing is a big dichotomy also from the analytical side. Think about taking imaging where you have to segment cells, stitch them, quantify versus sequencing based. Obviously, you don't have to take these steps. You directly read out a quantitative value per voxel or location that you then can analyze at different resolution scales. Another thing, really key ingredient we have been facing from both of these technologies is the integration with other modalities. We've seen many methods that utilize parallel single-cell atlases. We've generated in the field, for example, to map cell states or cell types, copy number states or other methods on these different modalities. And also histopathology is typically available and is being utilized to create atlases and integrate these data on different modalities. Now downstream, there's really a gazillion of different tools that each analyze and ask different questions, and I don't have really time to talk about all of them. I'll highlight a few of those. Think about cell-cell communication, think about inference tools, about how variation occurs in cellular spaces and other elements. And obviously, there's a huge and very, very active community to make these data accessible, visualize them in different elements on the web, in a tool, or combinations thereof. And I think that's been really critical to make the data accessible and utilizable as a community. So I want to start off with just highlighting one challenge. I think the previous talk highlighted this extremely well already. We have an abundance of technologies available. This list is ever growing and becomes more complicated and complex, and that's both a challenge and an opportunity. We have to pick from these technologies, we have to be able to integrate data and make them interoperable downstream if we use them, and this is often quite a daunting task. So I want to highlight here one part of the solution that we have been coming up together with a really large consortium and many people contributing to, which is spatial data. And the key idea is to try and represent diverse technologies using a sort of fixed set of primitives and asking the question, well, can we condense down the commonalities of these different technologies that we can store, handle, manipulate, and analyze these data in the end in the same way, which is a prerequisite to make progress. So the primitives are images, which is very clearly something we have available from histopathology to also other modalities, labels, which highlight where cells are, what they are, points, which could be transcripts, shapes, which is allocations, but then also the tabular views, which sort of aggregate information, count statistics, what we're really familiar with, we've been utilizing over decades now. And what's I think quite unique is that you can really bring those together and thereby represent a number of technologies in one way, then make data available. For example, here what's I think useful to say, under the disk, this really builds on the fantastic work of the OMI and GFF community that has created really high performant I/O methods, something I'll come back to later on, to be able to access data at scale. There's also really great work to interface those with machine learning approaches, make learning possible, and do all the exciting science that we care about later. So I just want to show you one example, how these primitives come about in the context of three canonical technologies. Visium, which has images, shapes, which are basically where are my spots, and counts, which essentially are my expression count tables for each of these spots. Very similar for Visium HD, just different spots, more spots, smaller spots. But then, for example, for imaging-based technologies like Xenium, we have this additional dimension of individual points, which are molecules. We have to first figure out what are actually cells, and then we can aggregate these molecules in these cellular niches to then think about quantification. And I think what's really quite nice to see, they can be leveraged across those elements and leave the commonalities between those. This has enabled, and I think we're just at the beginning of this, the creation of really multi-modal atlases, bringing together different technologies. Here you see an example of leveraging the best of the world's Visium versus Xenium. Visium is great because you have whole transcriptome counts, so you can do things like what Holger illustrated as well, copy number inference, have expressed genes at a resolution of up to megabase or up to 100 kb that allows you for genetic inferences. But then Xenium, obviously the right tool to look at subcellular resolution, cell state, cell counts, and you can now really create annotations, map objects onto the other, and start analyzing this data in a unified way. One other insight I want to highlight here at this slide is it has been very fruitful to separate the way we store data and what we do with analysis. And we hope this sort of comes up more in the technology and is more a common theme, that we need common standard how to represent data and then build all our tools downstream, that we can actually work with the same data and don't have to reinvent the wheel over and over again. So this is really a start of a journey, and then we see this starting already. I mentioned the spatial data being linked to other areas. Napari is interacted. There's a lot of questions about machine learning, and I've highlighted here also some of the names and groups who are currently contributing. It will be an incomplete list, but I just want to make the point that's really useful to anchor our efforts on common data, link them to visualization tools, but also link them, for example, to the R community, and we're really excited that this happens, that we even overcome the bridges between programming language and we agree on how do we store data, how do we manage data, and how do we make these data interoperable. And with this, I would like to sort of point a little bit towards where the future lies. 3D was mentioned. It's obviously a huge effort. Current 3D methods are based, for example, on slicing tissues in consecutive sections. Whether you call this 3D or not might be up for debate, but it's something we can now do thanks to the record-breaking decrease in cost from OpenST and NovaST and other technologies. And what this really entails is fundamental new challenges on how to store data and make them accessible. Here's, for example, that an effort that's really deeply rooted in the Napari community that has now utilized the OMI and GFF backend to be able to really do random access efficiently across petabyte scale data sets. You need to be able to aggregate across voxels, across different resolutions. And you have to do that on disk because you can't load any of these data sets to then being able to show these beautiful images and browsing in your data at different resolutions. And I think this will be an important bottleneck of making progress, of making data available, that we can actually store, process, and handle them. Another opportunity I want to highlight, this is a work we've been involved in. This is together with Dana Pear's lab and and Moritz Gerstrom, is cell segmentation. And I don't want to go into details. This is a different method how to do cell segmentation that leverages particular nucleus list transcripts and others and tries to assign them. I just want to make the point, it's not a solved problem. Yeah? How to segment cells is something we have to improve on. There's many challenges I highlight here that we also have not addressed, that we hope the community will make progress in. I just want to make the point here, there's really differences. Here's an example of a lung cancer data set with membrane staining, and you can really see that different segmentation methods give drastically different results. And in particularly, standard segmentations fail, and they struggle particularly also in areas where you have sort of inbleeding. You can zoom into certain areas, and for example, identify here examples of immune transcripts popping up in cells that are clearly marked as epithelial, and these sort of elements obviously complicate the analysis downstream. They make things very difficult for the analysis, and so getting segmentation right will be an important ingredient to making these atlases we're creating as useful as possible. I couldn't present here on the analysis if I were not mention the obviously hallmark questions of cell-cell communication. So inference of intercellular diversity and cell-cell communication is a really huge topic. You know, there's a whole sort of a tree of different methods that follow different principles. Most of them correlation-based, for example, looking at ligand receptor co-expression in space or non-space. And I think it's fair to say that we have only tipped the iceberg in how to make these methods work, despite their abundance. A really exciting opportunity, Holger mentioned multi-modal assays and multi-modal technologies, is particularly using those technologies, for example, to resolve really causally how a cell-cell interaction happens by expression, transition, seduction, and really being able to follow these steps bit by bit. So examples that I just want to highlight here is particularly looking at metabolomics. Liana is one tool that includes these elements. Niche Compass, also really nice work from Mo's lab that just recently came out that allows really to leverage these multimodal assays. And I think these are just examples of new analytical tools that are able to mine the information that we contain in multimodal assays and multimodal technologies as they come forward. Another sort of twist to this problem of analysis is there's no one-size-fits-all. There are specific questions that will only be relevant in some context, for example, disease. And again, I don't have time to list all of them, but just, for example, the example in oncology and other areas, we do have different processes we would like to map on it, spatial information, timing information of how evolution happens in a tumor, or infiltration processes. And I think we need to think about how these prophets work and how we can leverage these technologies. I think this sort of whole task of integrating spatial and non-spatial modalities is still not really quite solved. For example, now we have new types of labels we want to map, not just cell types, but also trees of evolutions in tumors, or, for example, trajectories that we can map on space. And I think this whole exercise of mapping trajectories is very important, can be quite useful. This is some work from Byrd lab, where we can sort of look at cell type abundance and look at clonal abundance, how they interact and other elements. So I think there's a lot of work to be done in that space, and think about how do we align data sets and how can we leverage this alignment. The last point, and here is really the sort of boundary of where we have solutions we can pick from, is a thing about diversity. It's a big element of leveraging diversity genetics, and due to the lack of any other examples, I just have here one example of what we've been doing before in single-cell assays, mapping genetic variants and asking how do genes change the expression level as a function of genetic background, which is something we will also obviously be able now to do in spatial data. For example, understanding genetic effects across cell types, or even continuously. Here you see an example of one of these beautiful single-cell EQTLs, where every dot is a cell. That's a cellular UMAP, not space, but we can now really map where do genetic variants exert effects, where do they change regulation. And we would like to do all of these things in space. And we will be able to do so leveraging large cohorts, leveraging the atlases that will come from this community, leveraging linear information. And I think that's a totally new area where I hope we will have also the technological answers to go about and the tools to answer these questions. And with this, I'm close to the end of my talk. I just have one last pledge, and that is we need to create and mine large spatial data collections. Here again, an example from the Omizara community, really think about these sort of tensor variate data sets where we'll be able to random access to any voxel, being able to pull those. We have to be able to store these data in cloud-based solutions very efficiently and mine them. Here's also one example from the Goldberg lab and connecting VTES to spatial data objects on cloud storage. Another example I want to highlight is, for example, from Fabian Theis' lab, and in creating large atlases to then learn foundation models and other elements, which will really possible only once we create these atlases. So I really hope we see more atlases, and this community would be in a fantastic position to create them. And with this, I would like to stop. I want to acknowledge people involved in this, and particularly the whole spatial data community. I hope I mentioned everybody here, and point you to the breakout that will happen after the session in room 2. And you see some of the questions we hope to answer, and I hope to see many of you there to discussing the future of spatial omics analysis. Thank you.
We are running a bit over, so I would suggest we leave the questions to the breakout afterwards, and we move on to the next speaker, which is Kevin.
Good afternoon, everyone. It's exciting to be here. First time I've been in person, but I've been connected with the HCA since 2020. Today I'm going to try to build off the last 2 talks to set us up for the discussions. And so one of the things that I hope to communicate today is while we're building atlases, ours in the upper airway mucosa, primarily focused on oral and craniofacial tissues, is that we're constantly having this discussion with our clinicians and our pathologists who are thinking much more at a global level, thinking about the patient, thinking about the tissues, maybe a biological sample. And then, of course, some of our basic science colleagues who are thinking about things at a molecular or cell level. And so we're constantly scaling our zooming in and zooming out, some of the ways in which we're talking about the data with our teams, which has kind of changed the way in which we have a vocabulary. We talk about tiers oftentimes, where they're performing a tiered analysis of the sample from a pathological tissue-wide approach, or whether thinking of it from a molecular perspective. And I think the last 2 talks highlighted this quite well, that some of the emerging toolkits in this space are making it possible for us to have that dialogue simultaneously. And so just as an overview, I've split this talk into 2, the goal of which is, I think, to really evaluate where we are as we were a community of bionetwork coordinators, now established champions, and a large bionetwork in which we were trying to build our atlas, which was a little bit late to the game. And so at the time in which we were assembling our first atlas, we had about 300,000 cells across health and disease. And we had tons of data coming in because many of those interested in our network wanted to perform spatial atlases. And so we decided to start to evaluate things that were commercially available. So at the time, we were looking at both probe-based RNA, spot-based, or seq-based RNA. We're looking at cyclical platforms for protein, but also non-cyclical. There's a number of technologies here. And we're not here to advocate for any of those, at least in this talk. And then, of course, there was things at a Rong Feng's group that were just getting started on spatial profiling of histone modifications in omochromatin. And so we had these conversations as we were thinking about this, and every time in which we came to a new technology and application as we wanted to establish our V1, or now we're calling it Vero 0.1, atlas, a new technology or methodology would either be released, open source, or it would be commercialized. And so I highlight just a paper that came out about a week ago that combined 5 modalities simultaneously. Many of you maybe saw this preprint that included a 100-plex protein plus single cell, not single cell, but spatial-based RNA, and of course, histone 3, K27 acetylation, and demethylation. And so for us, there obviously becomes a challenge for us to think about how it is we leverage the technologies we already committed ourselves to, and of course, downstream what's possible as we think about what atlasing means in each version, which I think in the breakout sessions we can discuss. We've also associated our group, HubMap was mentioned earlier by Martine. That original project never had any oral and craniofacial or upper airway data in it. And so all of our data we've been building out within our group became a complementary data set as a way to leverage both an HCA-facing approach, but also a HubMap-facing approach. And many of you may know that they have rooted some of their spatial analyses in a 3D reference atlas. Here you can see an example of that with the heart and some of the GI tract organs. And so from that, there's the ability to take your tissue, whether it's 2D or a preprint that's out there based on 200 micron thick sections of Merscope, and leverage those back into a referenced anchored site. And I'll talk about in our atlas how we're starting to do that with that team, which required some collaboration on both sides. Whatever the modality, whatever the thickness of tissue, whatever it might be, of course, there always becomes this challenge of linking together these assays when they're in the same slide, in thickness in 3 dimensions, to where they're overlaid and can be analyzed. And so we've tried to pursue this in our first approach. I'll show in just a few slides that we have, really as a way to think about some of these first assays that link back to our single cell data and become integrated with our spatial atlas. And so as a group, some of the early members of our team started thinking about what might be the kind of perceived challenges, rooting ourselves in version 0.1, and then thinking about what a V1 and a V2 that is complementary spatial and single-cell data together could look like. And so some of the things that we've identified that we'd love to expand upon with the groups today is that we know that sample quality is something that kind of becomes a dominant challenge for us. We never knew coming in from some of the archive samples that were biobanked of the upper airway mucosa, were they good? DV200s weren't successful in accurately predicting the outcomes. And so we know that for us, we had a decision to make on trying to evaluate what was appropriate quality moving forward. The same thing became a problem with us deciding between 2D and 3D, in which we're just now pursuing 3D, but wondering how it is that they can be compared and integrated with each other. And of course, the speed and slowness of these assays currently, while automation, I think, is dominating the single-cell world, manual processes still dominate and slow us down when it comes to spatial. A few other things that we've also noticed, and we've tried to link these modalities together, is that resolution can change. This adds another variable in terms of integration. We've had challenges in some of the complexity and size of the data. Reproducibility is a question. We've run the same sample a few times and seen different results. Again, getting back to that idea that the modality in itself is often dominated by a manual process. How the person pipettes on that day may affect the outcomes. And of course, standardization is needed if we're ever going to get this to becoming part of translation and clinical adoption, which is what part of our lab is rooted in. And then finally, I'll say, just as a final, some of the other things identified by our teams include the real need, I think, as we start thinking about scale, which was highlighted beautifully in the technology session. Scale and large-scale projects for us are going to be important as we start to make inferences with genetic diversity. And so we need harmonization with the platforms. We're going to decrease costs. And I think all of that kind of leads to this ability for us to include throughput as a dominant word for moving forward. And so our oral and craniofacial bionetwork is led by Anis Kiera and myself. There are 3 champions that we've named recently, and from that, we have a large bionetwork of pathologists, gastroenterologists, pulmonologists, but all types of oral specialists. And they all contribute to us trying to build what was really essentially version 0.1 for our group. In that time, because our group almost always has been funded by disease-focused research, where health is the reference, we have a number of data sets that have looked across the upper airway and even down into the lung. We haven't been able to avoid it. And so what I've highlighted here is that the group is simultaneously balancing both a single-cell tissue omics that has multi-modalities, also spatial omics, and some of our groups have even started to pursue the idea of spatial microbiomics, all this being linked to kind of a fleshed-out, fully formed version of an atlas. For us, there's also group members of ours because the saliva is easy to profile. Nasal swabs obviously dominated in the pandemic. This also becomes an opportunity for us to look at non-invasive sampling as a dominant form for us to link back to the tissues in which we're interested in studying. And so for our V0.1, we focused just on tissues. What you can see here is we were able to integrate about 11 studies, 3 of which were unpublished, so I think that was referenced earlier, is the generosity of our community. Those were donated without having linked publications to date. And what it gave us was the ability to look at 12 separate micro-niches of the upper airway, in this case focused on the oral mucosa. What's exciting about this is, again, focused on that disease research component of the oral and craniofacial bionetwork, is that most of them study a disease that shows up in one site. So there are parotid gland diseases that don't often show up in the other glands. There are mucosal diseases that show up not often in the other mucosa. And so this idea that was, I think, rooted again and leveraged by the Allen Brain Institute and this common coordinate framework becomes inherent to the design of the way in which we have our tissues. And so we are able to link them back to their niche, and so it gave us the ability to ask the questions about cell type and cell state relative to the position in which they're normally housed within the oral cavity. And so because we had not just the original single-cell data, but almost all these samples had spatial components and things in FFPE blocks, we're able to kind of build what essentially is our V0.1, which includes the atlas, not just single-cell, but also a multiplexed IF, parallel to a multiplexed MERFISH, and then also an integrated analysis between all 3. And so currently, just as a shout out, because Li Zhang, she's here, has done such a fantastic job with others as part of the integration team with Malta, Ellen, Lucia. V1 and V2 are being built out, and so our first atlas of about 300,000 cells is already being pushed to 1.5 million cells, 25 studies, many diseases now being included, and this also includes those biofluids as I referenced. And so this is we talked a little bit about the idea of ontology. So we start with ontology of structural cell types, meaning things non-immune, and things that are immune. And so you can see here just a representation of some of the data that we have. The goal of this, again, was a way for us to leverage the single cell data to design the panels the best we could based on the data that we had originally generated. And so what this gave us the ability to do on our multiplex IF, what you can see here as I laser point here, this is an oral mucosa. This is a salivary gland. We've run analyses spatially on a 40-plex antibody panel that links back decently, not 100%, to our single-cell annotations at tier 2. What you can see here is we can use a deep learning tool that we built called Tacit, the app called Astrograph, and from that we're able to look at not just the position of the cells, but also the context of their co-occurrence with each other. I can keep talking. So the co-occurrence with each other, but also in the context of the neighborhood analysis. So for us, one of the things we do is we build a three-dimensional space where we place all of the spatial data we generate and perform things like cytocommunity simultaneously on all the cells of all of the tissues simultaneously. And so for us, what this gave us the ability to do is to look at how the glands and mucosa were quite different, that were shared but also distinct neighborhoods within those tissues. I will also say that when we actually looked further, one of the things we could actually see is we could phenotype cells uniquely and found that, and maybe not surprisingly, that the cells that were immune, that were at the residency at the border near the mucosa, which is constantly burned, there's microbes there, it's challenged constantly. Those immune cells weren't just resident, but they were activated compared to the glands, which are deeper in the tissue. And so for us, one of the things that we then have moved forward on is looked at hub networks, where we can look at the neighborhood relationships to each other, and found that we can find these fibrovascular hubs, which are quite distinct from the glands that support a lot of immune cells within them. And so we're very curious about how these not only are established in development, but also how they change in disease. So if we also ask the question about mucosa, which we might think that mucosa are all the same, the head and neck cancer community tends to group anything that's in the throat or above as being part of head and neck mucosa. We can find different patterns of these hubs and cells and relationships the second we start bidding them out by their niche. And so in tongue and buccal mucosa, which is your cheek or tissue around the tooth, there's dynamically different rearrangements of both vascular and immune cells that we think have to be better appreciated. We even can look at the proportion of cells by these niches and find more innate or more adaptive cells, innate near the tooth, adaptive cells near the tongue. The tongue gets cancer way more often in these niches. And we think there's opportunities for us to better understand the disease application considering the niche and its origin. So then the last thing we did was we started noticing that the structural cell types, the non-professional cells, were doing a lot of signaling. And so what we did was we were able to look at the structural cell cytokine, effector cytokine signaling profiles. And from that, we built a MERScope profile that had 50 established receptor ligand pairs. So 100-plex of our MERScope experiment was focused on receptor ligand signaling. And from that, we could see that it's limited time, but all the cells of the single cell data matched a lot of the signaling patterns we saw of epithelia, fibroblasts, and vasculature as part of a structural immune axis. And when we dive in deep and look at this with ROIs, one of the things we can see, for example, at some of these epithelial fibroblast immune boundaries, we can see as we establish a ligand, a receptor, and then kind of a locked ligand pair within about 50 microns, we can start to find that there are hubs, irrespective of cell types. This is molecule only. And one of the things that we can find here are, as we start to establish some of these similarity of patterning of some of these chemokines, is that they actually sit spatially in distinct spaces within the homeostatic tissue, which we think are actually important for regulating kind of steady state biology over time, which we're further exploring. The last thing I'll say is that we've started to look even further down the line looking at homotypic and heterotypic cell interactions. And while it may not surprise the group, we're starting to deconvolve the concept that there are ways for us to look at homotypic cell-cell communication. These are all fibroblasts communicating right here near that epithelial border and the soft tissues around the tooth. But as we look further in disease contexts, these heterotypic interactions increase, but those don't miss also that there are a large increase in homotypic interactions, suggesting that there's something that's a compensatory role for these cell types to be communicating and regulating immune cues downstream. And so we are building with the HubMap reference, putting those data back, both the single cell and spatial data, rooted back to their original places of biopsy, so we can start to understand over time the anchored 3D positions and some of the effects from anterior, posterior in the oral cavity. And so it was mentioned, of course, we want to thank our teams. This is barely representative of the entire network, an incredible amount of support and collaboration, both domestically and internationally. We couldn't do it without these teams really over time. And what I'll highlight here, as Grace and I will be in the main hall, are some of the ideas of the scope, the challenges, and some examples of MVPs that we'd love to discuss with you as we build this into the HCA moving forward. So I thank you for your time and look forward to future discussions.
Thank you, Kevin. Again, we leave the breakout to do the questions, and we have as a next speaker Grace to bring the healthy atlases to the disease context for spatial atlasing.
Good afternoon. Thank you for having me here today. I think the talks today have already set up my talk. I'm supposed to try to convince you about why we should do disease, but I think many people have already brought up really excellent points about how normal is a lens by which we can interpret disease, but also how disease can help us to better understand normal phenotypes. So my name is Grace. I am a GIS BII fellow at the Genome Institute of Singapore, and today I want to share a little bit about our ongoing efforts at GIS as part of Dr. Sharon Prabhakar's team, where we've been looking at building a spatial atlas specifically for colorectal cancer. So colorectal cancer is the second leading cause of cancer-related death, both in Singapore and worldwide. And it is a very challenging disease to study because it is molecularly and clinically an extremely heterogeneous disease. And I don't think I really need to try to motivate why we need single cell and spatial to this audience, but I'm going to anyway, sort of to elaborate on how I think about things. So with single cell, we can dissect the diversity of the different cell types that form the disease, the tumor microenvironment, and so on. We can also use single cell to further our understanding about interpatient heterogeneity. So in this very nice study by my colleague Jonito in 2022, by taking out the epithelial cells from the single cell, subclustering them, what he found is 2 intrinsic epithelial subtypes, and that this could then be used to inform a better classification of CRC by taking into account the different compartments that actually contribute to the disease. But these are on two ends of the scale of heterogeneity. And another way to maybe think about it is in terms of spatial scale. So we have single cell on one end, patients on the other. But there's also cellular neighborhoods, or there's many different ways to call this. But essentially, what we want to know is the context that the cell lives in, what other cells it's looking at, the signaling and molecular gradients that the cell is in the environment of. And also tissue-level heterogeneity. And this is particularly important in cancer, where we have this concept of intra-tumor heterogeneity, which constitutes an important source of therapeutic resistance. And so, with spatial, I think, we have this opportunity to be able to start answering these questions. But at the same time, of course, these levels of heterogeneity also pose challenges in terms of how do we design our study. So at GIS, we've been, as I mentioned, trying to build a spatial atlas for CRC. So we took a large cohort across a diverse range of clinical and molecular subtypes. This work was done in collaboration with Dr. Ian Tan, Singapore General Hospital, National Cancer Center, Singapore. As mentioned, we had a single-cell RNA sequencing study, which helped to, inform a lot of our panel development as well as choice of samples. And then we applied to spatial omics technologies, one which is cyclic FISH for fresh frozen tissue, and then also 10x Xenium for FFPE, and this work was led by my colleagues Lipua and Eureka from GIS. We had to select these gene panels, and then altogether now we have over 9 million cells from 34 patients and 63 samples. I had to check the number. Okay, so I've tried to motivate why spatial technologies are really important for helping us to understand about spatial structures, but actually one of the learnings that we had on this project was that it's also really useful for cell annotation. So I think this morning we were talking about how you want to build evidence, and sometimes evidence across species or evidence from literature is really useful in defining your cell types. So this is actually a UMAP of our epithelial cells from our single cell. This is integrated across patients from 5 cohorts. In this little area are our enterocytes. And one question you might ask is, well, how did I decide the resolution of this clustering? That's always a very difficult question to answer. Usually it has to do with what kind of biological question you're actually asking. But we also had, in this case, our xenium data. So this is also epithelial cells, but xenium, you can also already see there's a slight loss of resolution. But we can use information from both, and by integrating both, okay, so the cell states here we can define are supported not only by subtype-specific markers, which would be like the more traditional way to build up evidence that this cell state is real, but we can also look at where they are in samples. So here is some normal colon, this is some tumor, and we can see that these different subtypes occupy the different spatial niches. So they have different markers, they have different spatial patterning. This gives us confidence that this is real. Once we have these cell types, we can then look at where they are in the tumor. So, but before we do that, I think we should look at where they are in normal so that we can understand what has changed. Well, and I'm sure the people from the gut atlas will know the colon has a very specific structure. So you have these crypts, basically, where you have the intestinal stem cells that live at the bottom. And then as they differentiate, they move upwards through the transit amplifying zone. And then finally, they start to gain specific absorptive or secretory functions up here in the mature cell zone. And we can see this very nicely in our normal adjacent samples that we collected as part of this study. You have this transition from more stem-like cells at the bottom to more differentiated cells at the top. Of course, if we take the analogous cell types and then we project them onto our cancer samples, we can immediately say two very obvious things. One is that, well, the very nice structure that we see in normal is completely gone. But I want to posit to you that these are non-random, that there is still some kind of like microorganization that's present within tumor that reflects the molecular and signaling gradients that have been changed in disease. But to be able to describe that, we first need some kind of vocabulary other than just cell types, but on the different spatial scales that lets us make these comparisons. So for these, what we did was we applied Banksy. So Banksy is an algorithm developed by my colleagues Vipul and Nigel. Nigel's also here and has a poster. And Banksy is basically an algorithm that unifies cell typing and spatial domain finding by allowing you to incorporate different amounts of information about a cell's neighborhood into the downstream embedding. And because we can create this embedding, then we can do other things downstream, for example, harmony integration to compare between disease and normal samples, which is, I would argue, like one view of the data. So Banksy is controlled by a parameter called lambda. So you have, when lambda is low, we're able to be in cell typing mode. This allows us to define what cell types are present within our sample. And then when lambda is high, we begin to define cellular neighborhoods. You can see here, they're just numbered by numbers. Naming neighborhoods turns out to be also even harder than naming cell types, I think. And then what we have in the end, is this like map where we have all these cellular neighborhoods and then the proportions of different cell types in them. I'm just going to highlight two. So one is this big cluster here, which represent all our epithelial high neighborhoods across normal and tumor. And the other, because I've been focusing on a lot of epithelial, we can also definitely find neighborhoods that are enriched for things like cancer-associated fibroblasts as well as immune cells within the tumor microenvironment. So if we go back to what I had described earlier, I had shown you a textbook picture. I tried to convince you that our data reflected that. We can also see this in the cellular neighborhoods. And I think why we need this vocabulary is because then it enables us to make comparisons, right? It allows us to say things, for example, that if we compare the abundance of different neighborhoods and disease versus normal, we see in this particular case that stem-high neighborhoods tend to be enriched in tumor, while mature cell neighborhoods are depleted. And because we have some information about what the normal structure is like, in particular about this differentiation axis going from the bottom to the top, the lumen side, we can also, for example, say that, well, we will anchor a trajectory that in this embedding space that describes transitions, basically in the proportions of cells within neighborhoods. We can see that very nicely in the normal, can project it onto also in the tumor. And then what we begin to see is this really interesting pattern where in some tumors, you have the stem-like neighborhoods that are high in stem cells, that are on the outside of these glands facing the stroma, possibly, we theorize, right, in contact with calves that are expressing certain ligands. And then the more differentiated cells, the more hypoxic cells, are actually on the inside of these glands. So that's kind of interesting. And I think it really shows us the power of like comparing tumor versus normal. I promised I'll talk about something other than epithelial, even though we focus mostly on epithelial. So this is an example of another neighborhood that we saw that is depleted in normal but enriched in tumor. And these are enriched in a particular cancer-associated fibroblast as well as endothelial cells and granulocytes. And we tend to see them on these lumen-facing sites. And what we think is that perhaps these calves are involved in granulocyte recruitment to areas of inflammation. Okay, so I talked a little bit about spatial neighborhoods, but those are fairly small. We can also talk about tissue domains and landmarks, and we work very closely with clinicians and pathologists on this project, and that's one of the ways that they think about these tissues. That is not, I think, like intuitive for someone like me who came from the single-cell transcriptomics world. And then they do this by looking at H&E. They'll take this and they'll segment it. They'll say, for example, this is a cancer domain, this is a stroma domain. This happens to be an invasive sample, so we also have the tumor normal interface here. But one thing that we can do now that we have molecular data is that we can use the molecular information to also begin to define these tissue domains. Of course, here I'm going to use something that we already know. In this simple workflow, we're going to take the density maps of the major lineages, segment them, define where the cancer tissue and the cancer-associated stroma regions would be. These would more normally be used to describe things like immune exclusion, immune infiltration, and also define where the tumor normal interface is for my invasive samples. And this allows us to do some interesting downstream analysis. So for example, we can calculate the density of different cell types in the cancer-associated stroma region versus the tumor region. We can also calculate the density of cell types with respect to the tumor normal interface. I'll just point out two interesting results. One is this light blue stromal cell type that tends to be enriched in the cancer-associated stroma, and then you can see here they're tightly wrapped around the tumor glands. And another is this epithelial subtype, this orange subtype, that again, we particularly clustered this one out also because we saw that it had a distinct location. It's not so obvious from the single cell alone, but what we observe is that it has this localization at the tumor normal interface. You can see here in orange in these samples along these lines. And then when we look even closer, when we go back to the histology, so this is these cells right here, look at them, and then you look at the histology of them. Turns out this is actually co-localized with regions where pathologists would describe as tumor budding. So now we found some interesting budding markers that are specifically are expressing these particular markers. And I think this is a really nice anecdote that shows how we can use spatial omics to integrate with pathology for histology-based diagnostics. So as a last thought, I said that we work a lot with the pathologists, and it's really interesting working with a completely different field because you get a completely different perspective. In the past year alone, I think 4 to 5 pathology foundation models trained on extremely large pathology data sets have come out. And these are imaging-based H&E technologies that are more or less disconnected from our molecular world right now, but I think they offer a lot of opportunities. So one thing that we have done is we've taken some of these foundation models, we've then extracted cell patches and embedded those, and then asked whether or not the embeddings produced by these pathology foundation models are actually predictive of gene expression, since that is one of the tasks that, of course, is really important to us in the gene expression world. What we found was when we compared the different foundation models, either trained on ImageNet versus pathology-specific ones, that if we use a simple KNN predictor downstream of it, then the pathology models actually perform better. But when we had a GNN predictor downstream of it, then sometimes the models trained on ImageNet actually perform better. And this maybe suggests this is still ongoing work, but I think this suggests to me that some of these pathology foundation models would really benefit from adding spatial molecular data to them as well. And as we go forward, we can think about it. But I think it's already very encouraging that we can build these models, and these are held out spatial omics data sets from our cohort. This one is the 10x data set that's released. But we can train these models, and then on a held out set, we can show that the model is able to actually predict expression of certain markers, in this case for epithelial, fibroblasts, and immune cells. So to close off, I've tried to describe some of the challenges and opportunities that have come about that we've seen through our work in using spatial technologies to study colorectal cancer, integrating with single cell to improve cell annotation, how to define cellular neighborhoods, describing that vocabulary, also defining key tissue domains or one scale up, and then finally integration with pathology. So this work could not have been possible without all the members of the lab, so Shyam's team. I will highlight especially Lipo, who is my co-lead on this project, our clinician collaborator, Dr. Ian Tan, as well as the pathology team from SGH and CCS, as well as the BII team and the AI research team. So I'm going to leave this slide up. Thank you very much for listening to my talk. I hope that I've inspired some ideas and thoughts for the breakout session.
Thank you very much. Let's thank all the speakers of the sessions again. So we're now going into breakout sessions for track 1, technology in breakout room 1, track 2, analysis, breakout room 2, and Atlas building and health and disease here in the main hall. We're a little bit delayed, so we have the keynote a little bit pushed to 5:45. So I welcome you all back here for the keynote later together. Thank you.
And follow Esther to the 6th floor if you want to go to the 6th floor.